Hello, folks. Here we are. It is Monday, January 4th. We're in our new year, our second half of the genetics unit. And today, we're going to take it a little bit easy as we start to roll back into getting used to class after having our nice long break. I hope everybody enjoyed their time off. For today, we're just going to do a little bit in the way of notes and a reminder and a little bit of prep, and then we'll spend any extra time we have making sure that assignments are up to date, that projects and labs are done, things like that. So why don't we get started? It's not going to be too strenuous, but there's a bunch of stuff that I do want to cover, and we're going to start by taking a look over here for a little bit of review. So we've already looked at all of these terms: dominant, recessive, genotype, phenotype, homozygous, heterozygous. I want us once more to make sure that we know how they go together and how they work. So we're going to start with the beginning, dominant. In this Punnett square, what is the dominant allele? Capital B. Remember, dominance will always be shown as a capital letter to show that nature they have of overpowering other things. Our recessive allele, therefore, must be little b. Recessives are always the lowercase because they tend to be overshadowed if a big dominant allele is present. All right, now what genotypes do we find in this Punnett square? Well, remember, a genotype is based on the genes. So when you take a look at this, we're going to be seeing capital B. That's one genotype. Capital B is a second genotype. Here's a repeat, and then little b is our third genotype. So there are all of our genotypes. Now, we haven't really talked about this, but what phenotypes would we see? Well, this one, and this one would all have the same phenotype. All three of these would have that dominant phenotype. Our example that we've been using is for eye color before break, so I just continued on with that here. We're looking at a capital B that is dominant for brown eyes. The darker color, skin, hair, or eyes are always dominant. And then the other one is our recessive phenotype, which we'd only see right here for blue eyes. All right, so once again, out of this table, this Punnett square, what are our homozygous examples? And remember, homo, once again, Latin for same. So here we're going to see capital B, or little b, as our homozygous examples. And our heterozygous example, and once again, hetero is Latin for different, will be capital B, little b. So this one is heterozygous. This one is homozygous dominant. This one is homozygous recessive. Good. All right, so we've got a little bit of review. Let's get into the new stuff. So we can look at scenarios and say, well, why do the alleles separate? How do we get all these combinations? And basically it comes down to two different rules. The first one is the law of segregation. Gregor Mendel came up with this forever ago. And basically all he's saying is that clearly we can each donate one of two different alleles. So they must separate so that we can get all the possible genetic combinations. So dad is heterozygous, mom is heterozygous. Here you can see the sperm cells and the egg cells. Each one only gets half of the genetic code. Half of 2 is 1. And then the second part, why can we get all of these possible combinations and not just one or two? Like, for instance, if this is I'm going to use this as my example for my alleles, and I am heterozygous, 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 heterozygous. Why don't my kids get just this set or this set? And the answer here is this idea of independent assortment. And basically what it means is that when a male body makes sperm cells, what it's going to do is it's going to pick one of these two A's, and then one of these two B's, and then one of these two D's and E's. But it doesn't need to pick the same letters on the same chromosome. So for instance, this first sperm cell right here might take the capital A and then the capital B and then the capital D and then the lowercase e. So it just swapped one out. We can have scenarios like the second one. As long as you have this genotype of these different letters, your sex cells, whatever they may be, can kind of randomly pick and choose which allele they're going to get. So a single combo can actually produce millions of possible different combinations, allowing every sperm or egg cell to be genetically unique. And that's really pretty amazing. All right, so let's start talking about the big thing for today, and that is this idea of linked alleles. So what happens is sometimes when we take a look at a chromosome, we see that there are two different alleles for two different traits that are really close to each other. In fact, they are so close that they're unlikely to separate. They're unlikely to get jumbled around when we take a look at independent assortment. The closer they are to each other, the less likely they are to be separated. All right, so we're going to take a look at one example here, and that is going to be our hair color and our eye color. All right, so here are our alleles. The eye color alleles, capital E is our dominant for brown, little e is our recessive for blue. For hair color, capital B, dominant allele for black hair, little b, dominant allele for blonde hair. All right, so we've got two different sets of letters. So when we take a look at dad's genotype and mom's genotype, we can see dad is heterozygous for both. We can see mom is homozygous recessive for hair, so she's blonde-haired, and homozygous dominant for eye color, so she has brown eyes, blonde hair. Now, what are all the possible alleles that their children could get? This gets way more complicated. All right, so first we have to take dad's alleles, and we need to separate them out, and then we have to go through and figure out every possible combination that we can possibly get. Then we need to get mom's and figure out every possible combination that might be in her egg cells. Now, for mom, in this example, it's very easy because it's it's going to be a little b and a big E. Those are the only possibilities. All right, then we need to make a Punnett square, but now we have 4 different possibilities from mom. We have 4 different possibilities from dad, which means we're not looking at a Punnett square like this with only 2 options. We're now going to be looking at a Punnett square that's much, much bigger. Don't worry too much about this. Oh, the arrows are off. This is just another way that I like to show to figure out the different ones, but you guys don't need to do that. They don't ask you to do this anymore. What they ask you to do is take a look at our Punnett squares from Hal. So the normal Punnett square has 4 boxes because there are 2 options. 2 times 2 is 4. But now there are 4 options times 4 options. 4 times 4, well, that's 16. It works the exact same way. You set dad's up across the top, mom's down the side, push dad's down, push mom's across, and you get all of the different possible genetic combinations. All right, so this looks like a lot. But now for the good part. The Regents doesn't ask you to do this. It asks you to be able to read this. And being able to read one of these Punnett squares after it's set up, that's much easier. So this is what they ask. You guys see this X sitting right here? That's the key. They will ask you, what is the genotype of individual X? What is the phenotype of individual X? So you just have to figure out X. So you bring capital B, little e down. You bring little b, capital E over. And this individual is going to be big B, little b, big E, little e, heterozygous for both. So we already have the genotype, big B, big E, little e. And then the phenotype, the physical traits, it's going to have the dark eyes, brown, and the dark hair, black. Done. So that's all you would be expected to do for these linked traits. It's really not that bad. It just looks scary. And this is why they're linked. I talked about it at the beginning, but here is one chromosome. You can see that X shape. This is all DNA. And here is allele B and allele E, right? And they're right next to each other. Here we have the two different sides. Dad's genotype, big B, little e, big E. And then they're just so close. They don't get separated much. They don't get swapped much. All right. So with that said, we hit a good point. I wanted to go a little further today, but I don't think I'm going to. I think we're going to stop there and give people time to kind of catch up and get back into the pattern for our classroom, for our school. So ladies and gentlemen, I hope that helped, made a little bit more clear that you guys recognize how to read one of these Punnett squares from hell and how they work. And beyond that, today, we're going to be working on catch-up. A lot of people emailed me over break about how, I forgot, I lost, I can't find. So today, we're going to give you a chance to get that done, to work on that. Tomorrow, we'll start up on some more new stuff, and we'll start looking into our next lab. So ladies and gentlemen, have a great day. I look forward to seeing everybody in class like always, and welcome back for the new year. May it be better than the last.